In this video, we'll show you how to set up and run an Invitrogen QuantiGene Singleplex assay in a 96-well plate format. The demonstration of this assay will be using cell lysates. Please refer to the how-to video for sample preparation for QuantiGene assays. Prior to setting up and running the QuantiGene Singleplex assay, calibrate the incubator or hybridization oven to both 55 and 50 degrees Celsius using our temperature validation kit. To properly set up the incubator, please refer to the how-to video or refer to the user's manual.
Make sure that you have a microplate reader capable of luminescence top reading. The assay is completed in 2 days with plate setup and overnight incubation on day 1, followed by signal amplification and plate reader detection on day 2. Prepare a plate map that includes 3 to 4 wells for background. Optimize the sample input using the video titled How to Prepare Samples and Determine Optimal Sample Input for Invitrogen QuantiGene Assays. Begin the assay by using an incubator or water bath to pre-warm the lysis mixture at 37 degrees Celsius for 30 minutes, followed by gentle swirling. During the same 30 minutes, remove a capture plate from 4 degrees Celsius and place it at room temperature. Keep the plate in the bag or pouch. If lysate samples have been frozen, remove them from the freezer. Thaw at room temperature followed by incubation at 37 degrees Celsius for 30 minutes. Following incubation, vortex briefly if samples are in tubes, or pipette up and down 5 times if samples are in plates. Leave the samples at room temperature until use. Prepare the day 1 reagents by first taking the probe sets from the freezer. Remove the blocking reagent from 4 degrees Celsius. Thaw on ice. Vortex briefly and centrifuge, and then store on ice. Use the assay table in the assay guide to prepare an appropriate volume of working probe sets for each target. Use the table in the protocol to calculate the amount of each reagent required. To make the working probe mix, combine nuclease-free water, lysis mixture, probe set, and blocking reagent. Refer to the volumes in the guide for 48 and 96 wells of the same probe set being used. Remove the capture plate from the sealed pouch. Vortex the working probe set and transfer working probe set to reservoir using a pipette filler. Use a multi-channel pipette to pipette 20 microliters of working probe set into each assay well of the capture plate according to the plate map. Here are a couple of tips and tricks. Use a single channel pipette if you have a limited number of wells on the plate. Do not scratch the surface inside the wells of the capture plate when adding the working probe set or the actual sample. Please refer to the user guide for detailed instructions and volumes for homogenates or purified RNA samples. Consider running different sample types concurrently on a single plate since day 2 of the assay is the same regardless of sample type. Add 80 microliters of lysate or diluted lysate to each well. The final volume in each well will be 100 microliters. For background controls, add 80 microliters of diluted lysis mixture. Make sure to change tips. Seal the hybridization plate using a day 1 foil seal. Remove the backing of the foil seal, center, and place onto the capture plate. Use a soft rubber roller or applicator to apply firm, even pressure to completely seal the plate. Make sure the numbers and letters on the plate are visible through the foil seal to ensure that all edges are properly sealed to avoid evaporation during overnight incubation. Centrifuge the capture plate at 240 G for 20 seconds at room temperature to ensure the contents accumulate to the bottom of the well. Place sealed capture plate into the incubator for overnight incubation at 55 degrees Celsius. Incubate overnight for 16 to 22 hours. Before leaving the lab, make sure the incubator temperature is stable for overnight incubation. Refer to the QuantiGene Singleplex user guide for proper instrument settings, and refer to the multi-mode microplate reader or luminometer user guide for setup and calibration procedure. Warm the pre-amplifier, amplifier, and label probe diluent solutions at 37 degrees Celsius for 30 minutes to dissolve any precipitates. Leave at room temperature until ready for use. Meanwhile, prepare the wash buffer according to the protocol in the user guide. Use nuclease-free water and wash buffer components 1 and 2. This volume is sufficient for one 96 well capture plate. To wash the capture plate, first remove the capture plate from the incubator. Remove and discard the day 1 foil seal. Have the previously prepared wash buffer ready in a reservoir. Add 200 microliters of wash buffer using a multi-channel pipette. Strongly invert contents of the wells into a sink or appropriate waste container. Strongly tap on a paper towel stack until no more new liquid can be seen on the paper towel. Repeat the wash step 2 more times with 300 microliters of wash buffer. Centrifuge the capture plate, inverted onto a paper towel at 240 G for 1 minute at room temperature. Using a clean reservoir and a multi-channel pipette, add 100 microliters per well of the previously warmed pre-amplifier solution. Seal the plate using a clear Day 2 plate seal using the rubber roller. Make sure to tightly seal the plate. Move the plate back to the incubator at 55 degrees Celsius for 1 hour. Transfer back any unused pre-amplifier solution. Remove the plate from the incubator, remove and discard the seal. Repeat the washing steps given previously, washing first with 200 microliters and then twice with 300 microliters. Always add the wash buffer to the wells before flicking and banging the plate over the sink. Centrifuge the capture plate, invert it onto a paper towel at 240 G for 1 minute at room temperature. Pour the pre-warmed amplifier solution into a clean reservoir and add 100 microliters per well using a multi-channel pipette. Seal the plate again using a new clear Day 2 plate seal. Place the capture plate back into the incubator, set at 55 degrees Celsius for 60 minutes. Transfer back any unused amplifier solution. Remove the plate from the incubator, remove and discard the seal. Repeat the washing steps given previously, washing first with 200 microliters and then twice with 300 microliters. For a full 96 well plate, add 11 milliliters label probe diluent to a 15 milliliter conical, followed by 11 microliters of label probe. Scale according to the number of wells or plates being used. Hold the working label probe solution at room temperature. Take the plate out of the incubator and then decrease the temperature to 50 degrees Celsius. Repeat the same washing and centrifugation steps. Add 100 microliters working label probe per well with a multi-channel pipette without changing tips. Seal again using a Day 2 seal and return the plate to the 50 degrees Celsius incubator. Incubate for 1 hour. At this time, make sure to take the substrate from 4 degrees Celsius and leave it at room temperature prior to use. Remove plate from the incubator and remove the seal. Repeat the washing steps given previously, washing first with 200 microliters and then twice with 300 microliters. Pour substrate into a clean reservoir and add 100 microliters per well using a multi-channel pipette. Seal the plate with a clear Day 2 seal and protect the plate from light while waiting for 5 minutes. After 5 minutes, remove the foil and the plate seal. Read the plate on a microplate luminometer, reading for 0.2 seconds per well. Do not add any mixing or shaking steps. Set up the plate reader such that the detector is reading from the top, as close to the plate as possible. Make sure that you set up the plate reader using the protocol before you are ready to read. For optimal results, read the plate within 15 minutes. Thank you for watching this video. To find out more information about QuantiGene Singleplex assays, visit thermofisher.com/QuantiGene.